Thank you. One usually thinks of the treatment of wounds and other things with antibiotics or the modern wonder drugs as something which is relatively recent. But actually, that's not the case, because the history on this goes back some 2,500 years to the early days in China. And at that time, we find that although the principle was not understood, that people were using such things as moldy soybean curds to treat boils and other infections. And surprisingly enough, the treatment was often successful, and it was successful because of the mold which was growing on the soybean curd. Now, the doctor applying these on the infection. Now, that was 2,500 years ago in early China. Coming back to the present, I went down to the library to find out something else about this field of antibiotics. I picked up the most recent text in this field. It's a newly revised edition of a book just off the press entitled Antibiotics, written by two University of California scientists, Drs. Robertson Pratt and John Dufresnoy. To make a long story, she's senior author of this volume. Incidentally, he is a professor of pharmacognosy, plant physiology at the University of California's School of Pharmacy, Dr. Robertson Pratt. Welcome once again to Science and Medicine.
Dr. Harold, it's nice to be back.
Have a chair.
Thank you. Well, this antibiotic field is getting to be really something these days. The antibiotics seem likely to become our most important weapons against many diseases, and actually, as more and more of them are discovered, it seems quite likely that in time we may actually be able to control most of the infectious diseases that plague mankind with them.
Well, perhaps the first thing we should do then is find out just what is an antibiotic.
Yes, well, the derivation of the word, of course, anti, against, biotic, from the root meaning life, something that's detrimental to life. As the term antibiotic is used in pharmacy and in medicine, however, it has a very specific connotation and is used to describe a chemical substance which is elaborated by one microorganism and which, when present, even in very dilute concentrations, is able to kill or inhibit the growth of other microorganisms. The antibiotics are biosynthetic. By that I mean that they are elaborated by a living organism in the course of its no-life functions.
Well, now, one thing I'm wondering about, we used to hear quite a bit about sulfa drugs. Recently, of course, antibiotics. Well, now, what is the relationship between these two?
Well, the sulfa drugs are strictly synthetic chemicals. That is, they're made in the laboratory by human chemists. They have a peculiar way of working. I have a diagram here you might get a look at. The sulfa drugs act by interfering with the metabolism of the pathogenic organism. Let's consider this as a complex molecule of a substance that is essential to the life of an organism. This substance is composed of 5 simpler constituents. Now, it so happens that the sulfa drugs are so similar in form to one of the constituents of this complex compound that at first the organisms are unable to discriminate between the two, and they incorporate the sulfa drug into this essential product. But of course, this is no longer the same product, and in due course the organism finds that it can't use this product in its normal life functions, and so of course it dies.
Well, that's fine for the sulfas, but now how about the antibiotics?
Well, that's a debatable point, but many of the antibiotics probably act in essentially the same way, but by interfering with other essential compounds.
Let's come back again to the soybean curds that we looked at a little bit earlier. Here is the culture of this material growing here, and here is the same material as it would appear not in culture. Suppose I take some of this now, and I'll just get a little bit of it and put it under the microscope here, and then turning on the lamp, we will see that this has a very beautiful arrangement. It's one of the penicillium group, and it's so called because of the fringe-like arrangement. It looks like a painter's brush, of course, and that's quite characteristic of that.
Yes, and it's this growth, of course, growing on the soybean curd that secretes the chemical, penicillin, which diffuses into the curd and from that, of course, into the wound of the patient. Of course, this would be a very unwise procedure for us to try to doctor ourselves in this way, because such a product is extremely crude and contains many impurities, some of which might be rather detrimental. Here is some penicillin in the purified crystalline form in which it is used today. Now, let me put some of this under the microscope for you. Now, if you look at this, you'll notice that the field is covered with some very beautiful crystals. which are crystals of the purified penicillin. As the organism is grown commercially, it looks something like tapioca of a large sort, and is it's the only sort, fungus as a matter of fact, the only mold which is the source at the present time of a useful antibiotic. There are many produced by other molds, but most of them are too toxic.
Well, let's step over to the wall here behind the lab table, and we have a breakdown here of where these various antibiotics originate. For example, here's the penicillin, of course, and it comes from a mold. That was the first one.
That's right.
And then another group, the actinomycetes, which they group an intermediate between the molds and the last group, the bacteria. Now, we've had a lot of these new ones come in here. What's the story on that?
Well, the actinomycetes have been the most fruitful source of antibiotics, of producing such things as streptomycin, aureomycin, terramycin, chloromycetin, erythromycin, magnemycin, a whole host of them. We shouldn't, of course, ignore the bacteria. Although they don't produce so many antibiotics that are useful, we do get bacitracin, tyrosricin, and polymyxin from the bacteria, which are extremely useful. They have a characteristic, of course, is their selective toxicity of these drugs. I think you have another chart.
Yes, that would bring out this point on this. In other words, the disinfectants, the thing that they do is to kill all of the microbes. And when we get over here to the antibiotics, we find that some of them kill certain microbes, others weaken some of the microbes, and finally there are some antibiotics that show no effect on others.
Yes, they are very discriminating in their action. This is one reason why antibiotics should not be used indiscriminately, say for the common cold, for example, because each one is able to do a specific job, and you shouldn't try to make it do a job that it's not capable of doing.
Well, I know all of us have had the experience of going to perhaps coming to the doctor's office and feeling that we have to have an antibiotic. We've heard about all these things where we say, Doc, may I have one? And he says, no. Now,
well, the doctor is actually serving in your best interest in that case, and you shouldn't argue with him, because the doctor knows which diseases and which organisms can be controlled by antibiotics. And he also knows that if he uses an antibiotic when it isn't really called for, he may sensitize you, so that some future time, when you have a really serious infection calling for an antibiotic, it wouldn't be possible to use it.
Well, let's step over here and see how some of these various ones vary in their activity. Penicillin, of course, is the first one that was discovered, and it works on streptococcal infections, most wound infections, and most bacterial pneumonias. And that is what this narrow-spectrum antibiotic. What do you mean by that?
Now, actually, of course, it looks as though penicillin covers quite a territory here, and it is useful in many diseases. But it turns out that most of these are caused by a relatively small group of organisms, and hence we call it a narrow-spectrum antibiotic. The next major antibiotic discovered after penicillin was streptomycin, which comes from one of the actinomycetes, and has a slightly broader spectrum, if you wish, but it's still relatively limited in its usefulness, and today is used mostly for the treatment of urinary infections, is effective in plague, and particularly interesting, is capable of controlling some of the very persistent tuberculosis organisms.
Now, how about these broad-spectrum antibiotics?
Well, these antibiotics, aureomycin, terramycin, chloromycetin, magnemycin, others that aren't shown here, have a much wider range of activity in that they can not only inhibit the growth of bacteria, but some viruses, some few viruses, some rickettsias. They're useful in such things as whooping cough, undulant fever, some of the dysenteries, some boils, some pneumonias. They're particularly useful in typhoid and typhus, particularly typhus, which some of our boys encountered in the South Pacific. Some few virus diseases. Because of this wide range, we call them broad-spectrum antibiotics.
Well, now, of course, a lot of these things do come from the soil, and I suppose now I go out in my backyard and I dig up some soil and I bring it into you. I know this has a new antibiotic. How are you going to prove it?
I thought you'd ask that. I picked up a sample from Golden Gate Park today. I'd like to bring it in the lab. Mrs. Tuck, this is Dr. Harrell from the Academy of Sciences.
How are you?
Mrs. Tuck is working on some soil samples now. This sample was carefully weighed out and then is placed into a dilution bottle of this sort, and you'll notice that you get a rather muddy, turbid solution. Now, actually, the soil is so loaded with organisms that if to try to culture these directly, we wouldn't be able to see the forest for the trees. There'd be so many colonies that wouldn't show well. So we're going to dilute this, and that's what she's doing now. She takes a small amount of this, puts it into a relatively large amount of water, and gets a much more dilute suspension, which will be useful for our subsequent work.
Now, how many times would she do that?
Well, it depends on the type of soil and amount of organic matter in it and so on, but we generally run from a dilution of 1 to 10,000 up to 1 in 100 million. You notice that she tilts this plate to get a uniform suspension of the distribution of the suspension over the entire surface. And she would run through several of these. She's making a second one now. Now these organisms actually are relatively slow growing, and after she completes this use of this pipette, which she can suck up the solution, and you notice again into the plate, and she will tilt this plate, because it's essential that we get uniform distribution of the suspension over the surface.
You know, the interesting thing here is that the soil, instead of being a reservoir of disease that's been considered in the past, actually is a potential resource for these antibiotics.
Yes, very true. For thousands of years, men have been searching for remedies to cure their ills. If you'll excuse a pun, in many instances, the cure lay literally at their feet in the soil. Now, these will grow rather slowly. They'll have to be placed in the incubator, but we have some colonies here, some plates, which were poured out a few days ago. I'd like to show you some of these. You notice this plate here, which has quite a dense growth. You can see a number of individual colonies.
Yes, there's everything on it. You couldn't very well study that, of course.
No, it's pretty difficult on a plate like this.
Which one would you pick now for study on it? Or
well, normally we would look for one of the actinomycetes because we know that's the best bet, but specifically we would look for colonies which are surrounded by clear areas, and we have marked one on one of the plates. This would shows that this organism is producing a substance which is preventing the growth of other organisms nearby.
Then you would take that out and set it up on another plate then?
Yes, we would pull out a colony of that organism, and it could be streaked out on a Petri plate. We would get something of this sort.
Well, I notice that doesn't grow all the way to the top. Does that indicate now that that's being repelled?
Well, you see, this is our soil organism across the top here, and we have cross-streaked with various disease-causing germs. Now, you're exactly right. This organism, which grows right up to the top, is inhibited markedly. It is not inhibited, whereas these, which grow only at some distance, are inhibited by the material diffusing from this soil organism.
Oh, the amazing thing to me about this is that what you demonstrate here very, very quickly had to be gone through some 100,000 times to discover just, say, terramycin or streptomycin alone.
That's correct, and these have come from soil samples gathered from all over the world, from the Philippines, the Orient, and every place else. Of course, this is only a small part of the actual procedure, as you said. When a useful antibiotic or potentially useful one is found, there are some 10 or a dozen specific tests that it has to go through before it can be released for human medication.
Well, how about some of these end products in the antibiotic industry that you hear so much about, B12 and some of the others?
Well, there are a number of byproducts. Actually, the antibiotics industry has caused a decrease in the cost of some other medicinals. Vitamin B12, for example, is very important in the treatment of certain types of anemia and extremely costly. Now it's produced as a byproduct from the fermentation of by streptomyces in the production of streptomycin. There are also other aspects of the antibiotic industry. They've aid in the growth acceleration, small amounts added to feed, stimulate the growth of young animals.
Well, we have some chickens over here. I think perhaps we'll demonstrate that. If I can hold these two little rascals. Both of these are the same age, and this one has been on a diet which included an antibiotic. You notice the difference in size. It's much larger. Whereas this little, I don't seem to be very capable of handling this. The smaller one here has been on just a normal diet. And there's quite a bit of difference in size. Now, this works very well on chickens, but it doesn't necessarily work on other animals. Recently in Steinhardt Aquarium, we conducted some tests using salmon. And adding antibiotics to the food, we found that everything happened that we didn't expect to happen. So finally, we had to take the salmon off the antibiotic. It didn't work with them, whereas it did work with the chickens.
That's a very important point. Earl, I'm glad you brought that up, because some parents might be inclined to try this on their children if they thought they weren't growing rapidly enough. It doesn't work that way. Actually, they have been used in a few cases of premature infants to prevent early weight loss, but on infants born at normal term or in older children, it's totally without effect and might actually be harmful by sensitizing the patient or the child so that at a later time the drug couldn't be used when needed.
Well, certainly this growth factor is very important, but the germ-killing factor is the thing that we're primarily interested in.
Yes, that's very true. The use of penicillin alone has reduced the case fatality rate in pneumococcal pneumonia from 75 to 85% in the pre-antibiotic years to less than 10% at present time. And in certain blood infections, septicemia and the like. Likewise, case fatality rates have declined to less than 10%. Of the antibiotics prescriptions written today, more than 25% of the prescription business in pharmacies in the United States is for antibiotic products.
One thing I'm wondering about, with all these various antibiotics available, how does a physician know that he should use this one and not this one?
Well, good question. The first step is to find out what the organism is and then to determine its sensitivity to the specific antibiotics. This culture that you're holding is a type of thing that might be sent in by a physician for determination. The first step would be to carefully remove this plug.
And to streak out the material
Streak the material out on the plate, like so. This plate contains blood, so that it provides the organism with a nutritional environment similar to that it had in the host. Now, this will have to be incubated for a few hours before anything would be seen, but I have here a plate which was streaked out yesterday, showing the type of thing that you might expect. You notice growth along these streaks.
Clear areas, those are important, are they?
Yes, you notice there along some of those streaks there are these clear areas showing that the organism has actually destroyed the blood in that area, and that's what it would do in a case of septicemia.
Well, now, with these various shotgun or broad-spectrum antibiotics that work on a variety of things, why is it necessary to know just what is involved by way of the germ?
Yes, the reason is that some of these antibiotics may be able to partially inhibit the organism without completely controlling it. And you may then want to go to another antibiotic in order to actually cure the infection. Now, if you've used the wrong antibiotic first, although you haven't killed the organism, you may have so altered its appearance that subsequent identification is practically impossible. Therefore, the only safe procedure is preliminary identification and later sensitivity testing.
Well, I see colonies on this plate, and they all look alike. Now,
yes,
there's only one type of bacteria.
You notice
there.
that they're all clusters of little round colonies that's showing that this is a staphylococcus. The next thing, of course, would be to take out a smear. I have made a smear on a slide. If you look at this under the microscope, you'll see these clusters of colonies that I referred to a moment ago. Now, following this, we would want to determine its sensitivity to the different antibiotics, and for that purpose, we would set up a series of tubes of nutrient broth containing the organism and containing different concentrations of different antibiotics. Where the organism grows, the antibiotic is not effective. The tube is quite turbid, such as in these. Now, where the tube is clear, it indicates that there hasn't been any growth, and in this case, it seems that we have two antibiotics which are effective.
And these two would then be
This is penicillin and this is streptomycin, which are able to control the growth of this bug.
Then the clinical laboratory would send the report back to the doctor who sent in this original specimen, and he would know what antibiotic to prescribe in this particular case.
And all of this takes only a matter of 24 to 36 hours.
Now, one thing that we've heard quite a bit about is this giving of two antibiotics at the same time. Now, what's the story on that?
Well, that's used because sometimes an organism which may not be vulnerable to a frontal attack, or only partially so, by one antibiotic, may have an Achilles heel that will succumb to a second, and also to reduce toxicity. Some of these antibiotics do have toxic properties. We have some fish that we've been working with in this connection, which probably show some of these symptoms. These fish have received a half a milligram of streptomycin, and these a half milligram of dihydro. You notice that they're pretty sick, most of them. Some of these are beginning to recover, but they're pretty sick. Now, that's because of the toxicity of those drugs. Well, in this case, these fish have received the same total amount, but only half of each drug.
And that way you cut down on the toxicity.
That way we keep the full antibiotic activity, but cut down on the toxicity of the drug, and this same practice, of course, applies in human medication.
Well, it seems to me that when we use the antibiotics, what we're actually doing is making the microbes work for us. In other words, applying one against another.
That's right. Like all living organisms, including man himself, the microbes have to have not only food but space in order to live and to grow and to reproduce. And the bacteria in the world of microorganisms do this the best way they can to defend their rights. They secrete chemical substances which are capable of making the environment untenable for their
Sort of a chemical warfare among the microbes, so to say.
Actually, you realize there's so many antibiotics that a dictionary has come out now, listing more than 1,000 antibiotic substances.
1,000 in this one volume.
Some 300 of which have been chemically identified. I think this is very hopeful for the future because many of these haven't yet been tested, and there's a great deal may come from it.
Well, one thing we were speaking of a little bit earlier is the effect of antibiotics on the general longevity picture. In other words, life expectancy on this chart here. I noticed that from 1940 or a little bit earlier when the first antibiotic was discovered, there's quite an upswing there now. What's the...
Yes, well, of course, life expectancy at birth in the United States has been increasing for the last 100 years or so and going up since 1900, due largely to improved methods of sanitation, health care, diagnosis, and so on. But there is a rather sharper increase since 1940 when antibiotics were first introduced. I think this can be put in a more dramatic way by pointing out that in about 1901, about this period here, according to the figures of the Census Bureau, of the children born in this year, 25% of them died before attaining the age of 25. But it is predicted on the basis of statistics that of the children born in 1948, that before that group has been depleted by 25%, the remaining 75% will be nearly 60 years of age. This is tremendous.
And of course, the antibiotics have entered this picture largely...
They're not entirely responsible,
no.
But have contributed to it.
The socio-economic aspects of this in the future are certainly something...
These are tremendous. They're a little beyond our scope at this
Well,
point.
Dr. Pratt, I want to thank you for coming to Science in Action to tell us about the battle of the microbes and chemical warfare in these groups. I'll be back in just a moment with the Animal of the Week. Our Animal of the Week is a hellbender. It's a giant salamander, in fact, the largest salamander that lives in the United States, and it occurs only in the eastern part of the country. And you'll notice what the distribution is. Patch up in here, down through here in the central part of the United States, and over in this section in here, and there's another type that lives here. Actually, we have two different types. We're not too much concerned with that at the present time, but let's take this down and look in the tank where we have the hellbender, and notice he's swimming down there. Very cute little fellow. I'd like to have you notice, first of all, that he has this ridge, or fold, fleshy fold along the side of the body. Now, that's very important in the respiration of the animal. When he's young, he has gills, but he loses those later. And this fold of skin has a lot of blood vessels close to the surface, so that quite often you will see these animals rotating the body like this, and that is to enable them to pick up more oxygen out of the water. That's the hellbender, and it's the giant salamander of the eastern part of the United States. It normally hides in the bottom of ponds and pools during the daytime and usually more active at night. During October and November, the females will lay about 300 to 450 eggs. They look like a string of beads. And occasionally, several females will use the same nest. Very, very interesting salamander. This is the giant salamander, the eastern part of the United States. 27 inches maximum length. And actually, it's small compared with the largest member of this family, and which is also the largest salamander in the world. That comes from Japan. It looks very much like this. Bob Dempster's bringing one in here right now, and I'm going to ask him to just put it right down on the... I'll get him right over here. Notice this fellow. You have to be careful with these because they have been known to bite. And I'll turn him around here and see if I can. Notice these bumps all over the head. Notice the spotting coloration. This is a small one because, according to some authorities, they reach a length of 5 feet, sometimes even 7 feet. Very difficult to hold on to. And one thing about this, it's good to eat. In fact, it's so good to eat that the Japanese government has declared this to be a protected animal, and no one is permitted to take them except for scientific purposes and for exhibition. This is the Japanese giant salamander, a relative of the eastern hellbender, which occurs in the United States. And of course, this is the largest salamander in the world when it's full grown. This is just little babies we have here now. Well, enough of our animals of the week. On our next program on Science in Action, we hope you'll finally be with us then. Thanks a lot.
You have just seen another in the fascinating television series, Science in Action. Science in Action is produced by the California Academy of Sciences under the supervision of Dr. Robert C. Miller.